Welcome to Brain Ponderings, your window into the world of neuroscience research and conversations with experts at forefront of their particular area of brain research or related topics. And today it's my pleasure to welcome Dr. Polina Schichkova. She's, well, where are you now, Polina? So you were at the Blue Brain Project in Lucerne,
Yeah.
Switzerland. Are you still in Switzerland?
Yeah. I am still in Switzerland, but I moved to Zurich and I work now in the company in the diagnosis in Proteomics field. But there I also do research and a lot of collaborations and internal projects as well. So yeah, I moved closer to there. A lot of data is generated.
Oh,
And before that, yeah, I was at the Blue Brain project at EPFL. And that is like the huge, or was a huge project by Henry Markram and many colleagues there that was aimed at computational simulation, the modeling of different aspects of the brain. And there I was doing my PhD in neuroscience, in particular the brain metabolism and its aging. So yeah.
what year did you receive your PhD there?
What year? Oh, it was 2 years ago or so, 2023. Yeah,
Okay, and
I
then
guess so.
you went,
Time flies. Yeah.
so you didn't do a postdoc, you went straight to the
No,
company.
I didn't do a postdoc. I went straight to the company. So the reason was more like personal. I wanted to stay in Switzerland and I had to find something very quickly. So I applied for both like the postdocs and companies, and the first place replied. I actually went there. Yeah.
Okay.
Um, but I am still still very happy with that decision. But yeah, it happened to be that the company was faster in replying than academia. So yeah,
Okay, and
even though I still keep the quite tight connections to academia, the collaborations and the stuff like that.
so I recently met you because um, yeah, I wrote a commentary for a paper you had in Frontiers in Science, which it's actually, it's the same organization, I guess, that does the Frontiers, but this is like a new effort um, of them. They have really a very machinery to review, and then to we did this online thing to go over this paper. I mean, essentially, and maybe you can talk about that in a minute, but it was really fascinating. You use this computational methods to take information on human brain, on metabolism, energy metabolism mainly, but related pathways, and then um, the electrical properties of neurons, and then integrated all this. Oh, and they also looked at different ages of human brain to try to get like a model of what's happening. It's a very complicated process, right? Because you got thousands and thousands of
Yeah.
biochemicals and pathways, and you got all these different neurons, and we can talk about this later. But I wanted to, before we do that, which is the main topic of this podcast, I'd like you to talk about you. So you grew up in Russia, right?
Yeah. And I moved to Switzerland to to do my PhD.
And
I did two masters in Moscow. One was applied mathematics and physics, and the other one was biotechnology. And both works were like in the bioinformatics related fields. And my bachelor's was in the applied mathematics and physics. And back then, I also did some internships in the computational drug discovery, computational drug design. So I tried quite different things, like exploring biology from different angles. But that was pretty much always computational, with exception of like a few courses there. I had some. kind of
Okay, and
non-computational
then
experience.
you learned a lot of neuroscience during your PhD
Yeah, yeah.
work at
It
the.
was also during one of my masters that my master project was on the autism, but again, the
Oh,
bioinformatics related
well,
aspect. Yeah.
that's interesting because there seems to be some metabolic problems in autism, maybe that are starting in early development, brain development in the developing fetus, actually brain. But okay,
Yeah, so
so
I went through like from development to aging.
so did I.
Yeah.
Yeah, I my postdoc was developmental neurobiology. I was
Yeah.
studying regulation and outgrowth of axons and dendrites and synapse formation. And then I got interested in cell death and aging, and because of glutamate, the brain's most important neurotransmitter. Yeah.
Yeah.
Okay, so w- let's talk about before we get into your modeling, it's important to kind of review. This is always something for someone who already has a neuroscience background. This may seem trivial or just kind of repeating stuff, but I think it's always good to think about the basics. So describe the different cell types in the brain and some of their interactions in relation to energy metabolism. Now that's a whole book there, but try to summarize it.
Yeah, yeah, of course, like I think most of us know, yeah, everyone know neurons, and they are different types like excitatory, inhibitory neurons, and then we can go even deeper into the morphologies, different morphoelectrical types and other classifications. But the neurons in general, they are the signaling machinery of our brain. They are what supposedly makes us think and create and all the other amazing things we can do. And then there are astrocytes that are very well known in the context of the energy metabolism of the brain as being cells which support in many ways neuronal function. But there are, of course, like more recent publications about more expanded role of the glial signaling, like glial cells, astrocytes are part of the glial cells. And this is, of course, like the very beginning of what we understand about this uh, complex cellular architecture of the brain. And well, there are the blood vessels, as you see behind in my background, and many other cells like microglia, which unfortunately I didn't get to study that much. But they are like well known in terms of immune related things. And again, like it's not the only function. And well, we can continue with the even less known cell types, but then yeah, uh, for me, these were the most important, the neurons and astrocytes and the blood vessels. And I think like if I had more time, we should of course include also microglia, which is
And
like
and,
super
and the
exciting.
oligodendrocytes that wrap around the axons and
Yeah, yeah. Well, axons is the other long story, which is even like more difficult than other cells for modeling, the computational modeling, because of their geometries, their properties, and because of how little we know about them.
so yeah, axons are literally a long story because some of them are very long. Yeah,
Yeah,
and yeah, it's you're right. It's hard to model because you've got so mitochondria, right? They're the energy factory of the cell, and
yeah, yeah,
the neurons are their morphologies are so elaborate, and there have to be mitochondria kind of distributed throughout the cell because the ATP that's produced, the general thinking is used pretty locally,
yeah,
although it
yeah.
can actually be released from the cells and act as a signal, which is kind of interesting. Um, yeah, so how do you go about modeling then? So you have to have some data. What kind of data sources do you draw upon that are available? I think my understanding is you're using a lot of
Everything.
freely available data
Yeah, yeah,
and kind of accumulating all this data and then analyzing it. I'm not
yeah.
good at computation. I can add and subtract and multiply and divide, but that's about it. So how do you go about,
So,
and I can do simple statistics.
yeah. I'm pretty sure. No, I was quite desperate in terms of the data search and the modeling because the data is essential for any analysis and any model. We cannot just base on nothing, on our ideas. We need to know what is measured there in the cells and basically the data, because the model I was building was very much the biology driven. So there are also like different types of machine learning, deep learning models. And there is actually the new virtual cell challenge, which launched just recently, where they aim to model the cells with more machine learning approaches. And it's a competition who predicts the best effects of perturbation and different contexts on the cells. Like very exciting and also thinking about it.
Well,
Um,
sorry to interrupt, but
yeah, sorry.
so is the company you're at using AI for anything? I mean, is it's
So what I'm describing now, this is like it has no relation to the company where I work currently.
okay, all right,
Yeah, this
all
is
right.
just something I saw when I was considering to participate in my free time, like because it's very relevant to my PhD project with this model, but very different methods, different angles, but kind of an intersection of both, I would say. But I can get back to this later and closer
I
to
just
your point. Like,
as an
yeah,
aside, my son he's working for a company kind of on the side actually. So he has a PhD in biochemistry, actually from Johns Hopkins. But um, these companies are, they're interested in the science now. I'm talking about the big companies like OpenAI and stuff. And so they're hiring people with a lot of knowledge of biology, biochemistry, and stuff to try to trick their model. In other words, come up with uh,
yeah, yeah,
things so that to try to reduce mistakes or what it's called hallucinations,
yeah, yeah,
I guess. So it's kind of interesting. These companies are hiring a lot of people to
yeah.
do this to help train the model. Okay,
Yeah,
so
so what data and what models was my project? So that was like very much the biochemistry and metabolism driven model because like every single enzyme I had to find in the literature how it works, what is measured about this enzyme, and also like about their substrate, their inhibitors, activators, like all these. I was like scraping the even like very old biochemistry papers as well as the new data sets because so the data are concentrations of all the possible metabolites and the proteins that we include in the model, and also the expression levels that had to be like the single cell, preferably. And that's also where the latest publications come as an input because that was like super recent data on the single cell brain RNA-seq that I included for the aging part, and that was like really the super important aspect to be able to model these aging processes in the
so that's from
brain.
so
Yeah.
there's yeah, I'm not up on all this bioinformatics work. So then that's from postmortem human brain. They
The,
do like
I was working on rodents, so that's like the rodent more mice brain model. Yeah,
Wait a
we
minute.
didn't have much data on the human at that time.
Ah, okay.
Yeah,
All right.
but I think
But there are, I mean,
many things are shared.
you can isolate RNA from using, there's a method called laser capture where you make a section of the brain that
Yeah.
can be, and then you essentially use a laser to extract the tissue and analyze it. Um, yeah, okay. Um, right. Okay, so, and there were like 16,000 some of these metabolites, is that right?
No. That's about the pairs, the interactions between the proteins and
Okay.
metabolites that we
So,
could analyze with this method. Because in the computational model, we can easily take any single or multiple elements, which are proteins or metabolites, and modify them and see the effects on the model. We could create this kind of the sensitivity map in young, in old, and then compare them, and then to see what are the changes, what are the what is more capable adapting to these perturbations. And that was like very interesting analysis, I think.
so
That's some.
how many metabolites then,
So,
approximately?
oof. Yeah, I get to remember now. It was about uh, more than 100. I would now need to check because it's been a
Okay,
while already after I
okay,
was working on this. It was a 158 differential equations, I think. And then the metabolites, like,
yeah.
yeah,
Okay, and then
I if I quickly check.
for the electrical activity, right? So neurons are excitable. So you have
Yeah.
all these uh uh molecules.
176, I got you the answer. And 151 differential equations. I removed some in the end. Yeah. And 176 metabolites with 95 enzymes. Yeah.
Okay. And then so you had that, and then you can look at um the essentially interactive changes or something like that.
Yeah.
Yeah.
So that is like super crucial for the brain and the coupling between signaling and the metabolism, that it's a dynamic process. And the very difficult aspect that the timings are different, like
Yeah.
electrophysiology is much faster than metabolism. And computationally, it's a big challenge for the models. Yeah, but I don't know like how experimentally with current methods one could capture simultaneously the electrophysiology on all the metabolic changes. I don't think it's possible now. Like, yeah, to measure this all online, like, yeah, and that's what we were
But
very interested in.
but different neurons have different firing properties. So for
Correct.
example,
Yeah.
typically the GABAergic inhibitory neurons they're firing a lot.
Yes,
They have high
yes.
metabolic rate, presumably. Other types of neurons may fire more slowly. So you,
So
yeah,
in this unitary model, we have like excitatory glutamatergic neuron. And we model with a relatively simple electrophysiology Hodgkin-Huxley type model with just some extras. And then I was also working on the circuit model with the different like GABA and glutamatergic neurons and their different morphologies and very detailed electrophysiology models. And then after I left, the colleagues continued and they actually published. I'm also a co-author, so you can find it. The paper in PLOS Computational Biology on the circuit. And there we have the different types of neurons. With the metabolism model is slightly adjusted between these two, mostly like excitatory, inhibitory categories. But the electrophysiology models are like extremely advanced in that part. So they're using this so blue brain stuff. And so basically the big difference in that part that allowed us to look at metabolism of the glutamatergic and GABAergic neurons was it was just like a few enzymes that they modified. I think it should have made it to the final model, but the important part was also the difference between layers, because we know from experiments there is a different density of mitochondria in different layers of the brain, and that was like a super cool addition to the model to scale the mitochondrial components according to the layers. And what we also know from the literature is the difference in the glycogen distribution in astrocytes. In the astrocytes, they are the main storage of the glycogen in the brain, and the glycogen is like the energy store, which is like not so much for what brain needs, but still helps when we cannot get energy from glucose, from ketones. We can have these stores. So these glycogen stores, they are also different by layers. I'm not sure if it made it to the final model, but we definitely looked at this with the earlier versions of this circuit. Like, yeah.
Yeah,
Uh,
I looked at that paper actually this earlier this morning here. It's pretty interesting. So the cerebral cortex there's kind of six layers generally considered. You go from outer to inner, and the whole cortex is like what, millimeter maybe or something like that. But these different layers have been ascribed to different functions or different roles here, and the circuitry. And what was interesting is that the middle cortical layers, like uh, 3, 4, and 5 or something, seem to show the biggest uh, vulnerability to energy impairment. And so I think it's layer four. I've got a lot of interest in stress, both bad stress, chronic uncontrollable stress, psychosocial stress, sleep deprivation, whatever, and then good stress, exercise, fasting, etc., keeping your minds active. And so with bad stress, that turns out the layer five, I think it's layer five, layer 4 or 5, cortical neurons have very high levels of glucocorticoid receptor, and they seem to be very prone to chronic bad stress. And similarly, in the hippocampus, the CA1 neurons, which are very vulnerable in Alzheimer's disease, uh, they have high levels of glucocorticoid receptor. And there's this whole literature on this, how uh, chronic activation of the glucocorticoid receptor is not good. And we actually showed that with good stress, the, well, it's a little complicated, but there's two receptors for cortisol, GR and MR. And with bad stress, GR stays up, MR goes down. With good stress, MR stays up, GR goes down. So it's kind of interesting. You can have elevations of cortisol, and depending on whether it's a good or bad stress, the brain cells respond differently. But anyway, aging, a lot of the changes that occur in aging in the brain, like general changes, increased oxidative damage to proteins, lipids, DNA, impaired autophagy, um, dysregulation of excitability, which seems to be actually pretty important just in normal aging, the ability of the neural networks to constrain their activity within normal limits is impaired or lessened, if you will. And then, yeah, so kind of the same thing happens with bad stress on a shorter time scale. So like bad stress for a month can cause these kinds of changes that take decades to see in aging. But with bad stress, the good news is it's reversible. With aging, well, we think we can slow it down, but reversing, nah, not yet. Hasn't happened yet. Okay, so in your Frontiers in Science article, and I'll put a link to the article and the online discussion of the article that was done in the description section for this podcast on both YouTube and the audio outlets. Okay, so you looked at these 176 um, proteins in young and old
Metabolites.
metabolites in young and old brains, and you did all these computational analyses. Can you talk a little bit more of how do you do it? So you didn't really describe in detail how you're
No,
actually
uh,
doing it.
yeah, we have it in the supplementary. It was
Okay.
a very iterative process. It was really like starting from bottom up, like starting from single enzymes and then modeling the single enzymes, then modeling the groups of enzymes, modeling
Okay.
the glycolysis.
I see.
See, like if the system is stable, then we are probably good. We can move on. And then like this, we could see where we should include some additional inhibitors, activators, because there are, of course, like so many signaling and regulation processes that it's like very hard to know what is essential and what can be
Yeah.
omitted. So
Yeah.
like this, it was like many years, like my PhD took 6 years and almost all the time was about building this model. And then like really towards the end, the last few months, I got it working. And then it was just
Wow.
like given that I included the RNA-seq data on the aged state as the scaling for each enzyme, and like a few things here and there that we described, like really minor modification, and then it worked. And like for me, it was a miracle because it took like almost 6 years to build this model. And then it was kind of like internal validation. Like it's not
Yeah,
real
yeah.
validation, but it was like
So,
that
so
for me
a
that,
lot of
yeah, like
maybe
it's
moderate
meaningful.
bad stress, and then all of a sudden relief from the stress.
Yeah, yeah,
Yeah.
so
And
true.
all right, so you're looking at things like metabolites involving glucose metabolism, fat metabolism. Uh,
Very, very little from fat, like
ah,
just a little bit on the ketones.
yeah.
The lipids are like way more diverse and complicated. We didn't include this pathway in the model, but of course it's like super important, and especially like lipid peroxidation and all these things. Like there is a lot to discover to add this to the models. Yeah.
Okay, so there's someone following up on what you've done, I assume.
So right now I don't know. Like the two things that we were finishing were this the circuit model and the application of this base model to the GLUT1 deficiency that is also published in the PLOS Computational Biology. The Jay Cogan is the first author, and I'm like co-first. So these two things were ongoing, and we finished them. And now there is no more Blue Brain project. There is the other uh, Swiss Brain Observatory kind of thing, Swiss Brain Institute or Open Brain Institute, and I am not aware of further efforts. I don't know.
Okay, but all this is
Yeah.
it's available on a website. Maybe
Yeah,
you can send me the
absolutely.
website. So
Also on
they're very
GitHub.
likely there's probably people all over the world starting to use
I hope
it.
so.
Yeah.
Once I got I was very happy once I got an email from someone asking me about the like how to run this model with the Jupyter Colab notebook, and I was like, someone
Oh,
is
wow.
starting to use it. That was super cool. I don't know if they like what will
Okay.
be the outcome, but I hope people will use it, and
What
yeah.
were what were your main findings, like take home messages? And then you had this interesting finding having to do with estrogen-related receptor. So could you talk about the
Yeah,
first, the
yeah.
kind of the general new
So
findings,
yeah.
or and then
So
that, yeah.
for me, like something new, according to my knowledge of the field, which is of course limited, and something new for me was this flexibility changes with aging, that our brain becomes kind of less adaptable, like
Yeah.
that if we make some perturbations, it can make more hard, like it's worse in the age system, while in young system the effects of this perturbation kind of gets distributed in the network. And that was like something super interesting for me. And then the ESRA, like I actually like was searching the like what is this doing, like is it relevant, and it seems to be relevant. Like it was really interesting. Like we went to this sensitivity analysis, and then we found the most vulnerable enzymes, and then we looked at the shared the transcription factors with the transcription factor enrichment analysis and found this protein. And then it also interacts in with many aging related and longevity related proteins like sirtuins, histone deacetylases. That was like very interesting. I don't think it's been shown before.
No, no, no.
Yeah.
Yeah. So what that identifying that estrogen-related receptors seeming based on your modeling to be important in brain aging, then other labs can start manipulating
Yeah. Hope
that
so.
gene in, I guess, whole animal. I don't know, maybe that's already been knocked out. I don't know, but and then models of brain aging, age-related neurodegenerative disorders. That's another thing people could do with this aging. It's everybody knows, I think, is a
Yeah.
major risk factor for Alzheimer's disease, other kinds of dementia, Parkinson's.
But that's also interesting for me, like at which conditions, like what is the crucial difference at the molecular level between the
Uh,
healthy aging and the not so healthy aging? Like that's, I think
yeah, I don't,
we are missing
it's
a
hard
lot
to
of
say there's a
information.
like one protein
Yeah, yeah,
and
for
gene,
sure.
but we do know that with aging, the ability of cells to respond adaptively to stress is compromised. And that's something I've been interested in a long time, that uh, how cells respond to stress in good ways, and what happens when they can't respond to stress well. And that definitely happens with aging at the cellular level and whole individual level. The stress, what it doesn't matter too much what the stress is. I mean, it can take like heat stress, right?
Completely.
We have global warming and these episodes of very high temperature, unusually high, um, and people are not adapted to. Yeah, it's going to be interesting. Like if in the United States, there's some states in the Southwest, Arizona, for example. Right now, I have some relatives living there, and it's like every day it's above 110 Fahrenheit, which is about, what is it, 40,
That's a lot.
It's about 40 centigrade Celsius, I think. And so everybody's in air conditioning all day. And so what happens if there's like a big power outage? They're not adapted to heat, right? Because they haven't been exposed to it. And yeah, and people are more out of shape than they used to be, don't get enough exercise, and overeating makes you more vulnerable to heat. So I'm just using heat as kind of a
Yeah,
very simple example, but other kinds of stress too. And the stresses with normal aging, so there's oxidative stress. Uh, Neurons in the brain seem with aging not to be able to take up and use glucose as well. And that's definitely
yeah,
true in mild cognitive impairment and Alzheimer's. And so this kind of flexibility, it's
yeah.
the idea of flexibility. So we can talk about metabolic flexibility. If the neurons with aging are not flexible in what they can use as an energy substrate, like if for some reason glucose availability is reduced somewhat and they can't use fats, then they're much more uh,
Vulnerable.
vulnerable. Yeah. So the flexibility thing is really interesting. You demonstrated at the level of these metabolites, but
Yeah.
it also
What's
applies different levels on up.
What's interesting that we saw that it changes differently between neurons and astrocytes, and the differences were like mostly driven by these expression scaling differences that we applied. And that is what was crucial about having single cell data, that neurons and astrocytes, they don't age the same way. And even though they have like many enzymes are the same, they are at different levels, and their changes with aging are not proportional for many of them,
Yeah.
and also for metabolites. Yeah.
Yeah, it gets very
So it's
complicated. So the
yeah,
astrocytes,
very so
electrical excitability is one major difference, but there's a lot of other differences between
yeah.
neurons and astrocytes. But just at a general level, neurons are exposed to much more stress. Astrocytes can be replaced during aging. Most neurons in the brain, there aren't any stem cells to replace them. There are in a couple brain regions, but in most cases not. So your neurons are around for your lifetime for the most part. And astrocytes, they can die, and then there's stem cells that can replace them, or they can proliferate themselves under the right conditions of the astrocytes. Um, yeah, so it could be something as simple as that, that there's like turnover of astrocytes and kind of rejuvenation of um, the population. I don't know.
Would be interesting.
Yeah,
Yeah.
yeah. Um, okay, let's see. Right. Um, what else? Is there anything else based on that came out of your work that take-home messages or things that weren't in the Frontiers in Science paper?
No, we
Yeah.
described pretty much everything, and
Okay.
that was a lot. Yeah. One thing we didn't really mention here today was also kind of a validation that it's meaningful. Well, of course, we had like more formal validations that are like boring things to me that we compared to some literature and so on. But one thing was interesting was when we tried to model potential interventions that would help to counteract aging. And these potential like anti-aging therapies ended up in the very well-known interventions that are like diet and exercise. And that was like quite motivational for myself to, I mean, I was conscious about these things, of course, but to be more kind of data driven in my
So you personally
lifestyle
are
choices.
exercising more.
Yeah, yeah, I do, and now I like think like, yeah, yeah, I need to go running. That will help.
Yeah,
Yeah,
definitely.
that is like uh,
Well,
will
and
drive some lactate and
that is kind of validation of the model when that comes
yeah.
up.
And this is so powerful that this was comparable to the very targeted therapies acting on the multiple enzymes in a very selective way. That is very cool to see that simple things we all know work so well, but they are simple in concept, but quite difficult in implementation with all the modern lifestyle, of course. And everyone is, not everyone, maybe
Well,
like many people would be
especially if
like
you're a
happy.
computational biologist, right? 'Cause you're spending a lot of time sitting, most
Yeah,
of your
yeah.
time sitting at the computer. So are you starting to like get up every hour and walk around and things like that?
That not so much, but I started to run like almost every day, which is like a
Good.
big thing for me.
Yeah, yeah.
That was a, yeah.
Yeah, usually if you can stick with it for a few weeks or a month, then you'll miss it if you can't do it.
Yeah, yeah,
Yeah,
you get addicted.
yeah, yeah. So that's
That's,
activating the
yeah,
adaptive stress response pathways in a good way. So your BDNF levels are going up. So
yeah,
what? So your data then, I mean, there's huge databases on BDNF, for example, a huge amount of information on BDNF. It would be interesting to so is it set up so like if I was interested in how BDNF signaling, which the pathway is well known, receptor, track B, and downstream pathway, and genes that are upregulated by BDNF, and uh, it's an activity-dependent neurotrophic factor. So you'd think it, and there's quite a bit of evidence it's important in energy metabolism. For example, we showed that BDNF stimulates mitochondrial biogenesis in neurons, a generation of new healthy mitochondria. And so it seems like it'd be interesting to plug that one. Can that be
yeah.
plugged into your system and say, okay, if I were to increase levels or activation of BDNF pathway, how would that affect with aging the flexibility, decrease in
Yeah,
flexibility or
yeah,
something?
exactly. That is possible. Like that's the whole point of this
Yeah.
uh not black box. Like machine learning in many ways is a black box model. And this is this type of modeling that I did is very transparent. So we can include new pathways, new signaling mechanisms, and then we will see the effects, the downstream effects on the entire system, and we will know like what is changing. And but to include these, we will need to add some equations basically that describe what are the missing enzymes, missing signaling
Hmm.
molecules, proteins. Yeah.
I see. Wow.
It
Yeah. So that's
depending on the mechanism, depending on the pathway and so on. Like it can either be relatively simple or it can be difficult. It would really depend on case. Like for the GLUT1 deficiency, it was relatively simple. It was, I would say, it was simple because we knew like what is happening in the GLUT1, GLUT3. They were already there.
so
So
this mention so GLUT1 and GLUT3 are glucose
yeah,
transporters.
glucose transporters. Yeah, yeah,
GLUT1's in the yeah, it's in vascular endothelial cells and it gets glucose into the
yeah.
brain, and then GLUT3 is in the neurons and it gets the glucose into the neuron.
Yeah, yeah.
So the GLUT1, so there's GLUT1 knockout mice, right? GLUT1
Uh,
deficiency. So GLUT1 deficiency in humans will cause some neurological problems, right?
yeah, 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 and that's also like with some seizures. I think the
Okay.
phenotype. Yeah. But again, I was mostly on the modeling part rather than the deep into biology. So I wanted to know the molecular pathways and what happens, how we implement these changes
Yeah.
at the molecular level. Yeah, but overall, I think it's something like seizure phenotype. But I might be wrong. I'm not expert in this particular thing.
So
But
if you
yeah,
happen to have
my
GLUT1 deficiency, you would want to increase your ketone levels to suppress the seizures, I would think. It'll probably
yeah.
work.
Yeah,
I mean, it's used for epilepsy patients, ketogenic
yeah,
diets, and actually intermittent fasting works as well.
yeah, yeah. So we
Yeah.
tried all these different things and we're looking at which work better and which work not as good and like basically changing the firing. And yeah, so in that case, modeling the GLUT1 deficiency is relatively simple because you don't need new pathways. But
Yeah.
then you want to include the new pathways.
Yeah.
That's going to be more complicated because you need to balance this model. You need to know your pathways very well. You need to get the data. So, but the main goal is that the model can be modularly expanded to address all these specific questions, and then we can learn what happens in these different conditions. So that is
Yeah. So
very exciting.
you integrated the model of electrical excitability into it, and yeah. So the next logical thing would be to look at calcium signaling, which in itself is kind of complicated, but still, that's calcium influx is like the most important signal
Yeah.
for all the consequences of neural activity.
We
Uh,
have a little bit of calcium in the model, yeah. Like
yeah.
not super advanced, but a bit of calcium signaling. Yes, it's present there. And also endoplasmic reticulum
Ah,
stores of calcium. I've been looking into that, like how we can use that thing. But again, this is also like quite complicated aspect as well. Yeah, and absolutely important for sure.
and the company you're at doesn't have anything to do with neuroscience, or what's the? I'm just asking a question. Does it?
Yeah, yeah. It's doing proteomics, and I think I'm in the super unique position there. I get to work with the very high quality data on all different aspects, like that's mm, including neuroscience. Like I cannot say too much because I don't know
Yeah.
what I'm allowed to say and what's not.
Yeah.
But from the what we already presented, we did have the project on aging on the proteomics data, and that was presented at a few conferences, and also I presented it at the HUPO conference. And then also I got to work with some cancer data. So very interesting, very different projects. And that's not computational modeling, but just like the data analysis, the statistics, so
I see.
these classic
Right. So
things there,
they're taking like blood samples and measuring or whatever source,
whatever. Yeah.
and then
My,
quantifying levels by, I guess, mass
yeah,
spec analysis of hundreds or hundreds and hundreds of proteins.
thousands,
Yeah,
thousands. Yeah,
thousands of proteins.
my work is mostly in collaborations and internal projects, like all the fun stuff
Oh,
that gets presented at the conferences and in publications, so that people can see what they can get with the data if they send samples to the company. That's
oh,
kind
I
of,
see. I see. Yeah,
yeah.
yeah, yeah.
So even though I'm not in academia anymore, well, maybe I will be back at some time. I don't know. The
I
future
think
is
a key
unpredictable.
thing
I
there
still do
would be,
research.
advancing the methods so that, like, kind of by analogy with DNA sequencing, right? It used to cost a lot of money
Yeah.
for, sequencing one person's DNA or something, but it's
Yeah.
getting less and less. And the proteomics is also, at least we did some proteomics and sent it out
Yeah.
actually to a facility. And it was quite expensive. So I think technology development will be important in that
Yeah. Yeah.
too, making it easier to,
And actually, like, I'm like, that's not related to the company, but my personal feeling is that in context of this computational modeling and understanding the molecular mechanisms of the cells function, this would be super cool to have the proteomics single cell data, because I've been using the RNA-seq data, and there is, of course, translation, degradation, all these machineries that make the difference.
yeah.
And sometimes the for some, or I don't know how many, I think for many proteins, their real levels in the cells are not the thing that the
Ah,
expression tells us.
yeah,
So
yeah,
we
yeah.
might see like many new findings once we get such data.
Yeah,
But maybe it's there now, since it's been already quite some time. At the moment I've been working on the model, I didn't find such data, and I was like,
yeah,
okay, I use RNA-seq. Yeah,
yeah. Huh.
yeah,
Okay. And right. So one thing in your paper that with aging, so you looked at young, I can't remember. You had
young
young
and
and
old, and
old.
it was binary.
It was
Yeah,
binary, so you didn't really
there
have
was, yeah.
individuals through the whole spectrum.
No, it
So,
was binary. It was more like the fold change of the RNA-seq data. That's the main driving factor.
well, if you look at function of the brain in relation to aging, uh, memory ability, and also like ability to switch attention and, like, I guess, quickly switch attention from one task to another. That declines beginning in the 30s and 40s, and then it kind of goes down kind of in a linear manner, and then like when you get into my age, close to 70, then it's like that. So yeah, I guess my point there is in thinking about aging and the fact that I don't see us reversing aging. Maybe you can slow it down later in life, but ideally you want to start early and doing the thing,
Absolutely. Yeah.
yeah, and keep your brain active and your body, and don't eat too much food, particularly junk food. And
As like with anything, the prevention is
yeah,
easier than reversing the damage.
yeah, and education, like right now we're doing, right? So that the healthcare system in Europe, and at least a lot of countries, is better in many ways than the United States. At least there's some uh, emphasis or efforts to promote prevention and risk reduction. In the United States, it's all for profit. everything. Like the whole healthcare industry is for profit, and the company, big agriculture, and the companies that make the unhealthy food, they're there's no put hasn't been any pushback in the past much against them, and the pharmaceutical companies don't really have much interest in prevention or risk reduction, except with a few things, I guess, like the statins to lower blood cholesterol, or maybe, but still, and those things, you're targeting one specific thing, but you're not targeting aging per se, and so yeah, you may be able to reduce cholesterol, but everything else is still progressing downhill. Yeah, so education and early lifestyle changes. And then another thing in the United States, we get this what I can call transgenerational propagation of unhealthy lifestyles. So if you grow up and your parents don't exercise and they eat junk food and they watch television all the time, don't do a lot of intellectually challenging things, then you as their child are more likely to adopt that those lifestyle habits, if you will, and continue them. Well, not necessarily, but just in general, that's true. So then you get kind of this propagation of cross generations of poor health. So that has happened here in the United States, particularly in the last 50 years or so, 60 years. Yeah.
But I think like now with all the social media and everything, the awareness everywhere is increasing, and yeah, it's becoming popular. I think maybe it's just in my bubble.
Hopefully, yeah, you may be
Um,
right. Maybe I think you may be right. The younger people, hopefully that's a general thing that's happening with the younger people. The social media, though, has also caused a lot of problems because there's a lot of misinformation or
yeah,
disinformation.
yeah.
Like, for example, even on YouTube, it's amazing, because you go on there and a bunch of videos will pop up, right, based on, I don't know, these algorithms or whatever. And then some of these, like the title there's one I saw the other day, more than half of, this is what it said, like the headline for the video was, more than half of China flooded, millions of people dead. And
No.
that is just, no, that's
Yeah,
that's
yeah.
an extreme
Yeah,
example,
we
but
need the critical thinking to understand what's
yeah, yeah. So,
good source of the data and
but
what is not.
there's more subtle
Yeah,
things, right? More subtle things.
yeah, yeah. True, true.
So my podcasts don't get so many views because I don't do all this sensationalization of what we're doing. Uh, okay.
Yeah. Well,
And I don't have any ads on mine either. So I'm not making, some people, they apparently make money from letting the companies put ads on your YouTube or Spotify or whatever. So I don't do that. That's a waste of people's time. Okay, anyway, um,
yeah.
we've been talking almost an hour. So, Polina, thanks for taking the time for this. Kind
Thank
of
you.
take-home message is there's a huge amount of data on the brain at the molecular level, at the metabolism, energy metabolism level, at the structural level, the organization of the cells, movement of different metabolites between cells. We didn't really get into like
Bad and transport. Yeah.
the lactate, which can be generated in astrocytes and used as an energy source by neurons. and the glutamine cycle. So
Yeah.
glutamate's an amino acid, and when it's released from neurons, it's taken up by astrocytes and converted to glutamine, which can be involved in energy metabolism. And then the glutamine can go out and get into the neurons, be converted to glutamate, and then GABA is produced from glutamate. So there's an enzyme called glutamic acid decarboxylase that converts glutamic acid to GABA. So that's really interesting shuttle system because you
We
got
included those things. Yeah.
yeah.
Just, just yeah. There.
Yeah.
Yeah.
Okay. So
That's
in the future, but I think the artificial intelligence, I think that there's a lot of potential for it to help with
I
this
think
somewhat,
like
as long as it's
personally,
done right.
yeah, I think the very promising and exciting application is a combination of both, like including the neural differential equations, for example, where we know like there is some part from the biology, some mechanism, and then for those parts that we don't know, we include these machine learning, deep learning components that we can learn from the data. So even though we don't know how exactly something functions, we can learn this thing and we can include those parts that we know from our textbook knowledge. So that is, I think, like very exciting application, like taking the best from both worlds, this biochemistry
Yeah.
and detailed bottom-up modeling and the AI and machine learning. So
Yeah.
that is,
And
I
then
think,
ultimately, deciding what's the most actual real world experiments to do based on the
yeah,
model,
yeah,
right? Because ultimately one has to confirm it
of course,
through.
yeah,
Yeah.
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 The experimental validation is
Yeah.
crucial, of course. Yeah.
Yeah.
And I think the models should help experimental people to, it should guide them to know where to look, like which
Exactly.
note down to do, like, yeah.
Right. Right. To narrow down what's the most
Yeah. Yeah.
likely, the most probably, the highest probability of, you manipulate gene X or protein Y
Yeah. Yeah.
and it slows down aging, y- you know,
Yeah.
to,
Exactly.
to kind of, yeah, distill it down. Okay. Thanks, Paulina. I'll let you get back to
Thank
work on
you.
your proteomics.
Thank you. It was a pleasure to talk to you.
All right. Bye.
Looking forward to your papers to read them. Thank you.
Okay. Bye.
Bye. Thank you.